Okay, I think my iPad deleted the video that I just made for you guys, so let's try that again. Let's talk about the cascade that you guys learned in class. Whenever your cell undergoes what we call cellular stress, and the way I think of this is just DNA damage. So remember, DNA is the instructional code of your cell. If your DNA gets damaged, you are messing with the instructions of your organism, and you don't want to pass that on, right? If your instructions are broken, you want to contain that damage. Otherwise, if you keep replicating on broken instructions, you can propagate a mutation, and you can cause all sorts of bad things to happen. So whenever your cell undergoes cellular stress, a protein called p53 is generated. p53 is a transcription factor. And remember, transcription factors will bind to enhancers or silencers, but in this case it's an enhancer, which will encourage RNA polymerase to bind to a promoter and transcribe whatever gene is in front of it. In this case, this gene is p21, the protein. So by activating, by being the transcription factor for the p21 gene, p53 is responsible for making p21 by being its transcription factor. Cool. Now you have two proteins floating around. You have p53 and p21. This one's constantly making more of these. p21 is actually an inhibitor. So that's what these arrows mean. This means inhibit or kill, stop. This means go or upregulate or make more of or something like that. So p21 inhibits this complex. I'll do it in orange. It's a cyclin CDK complex. So what is CDK? CDK is a cyclin dependent kinase. Alarm bells should be going off in your head saying that kinase tells you it's an enzyme. Kinases specifically are enzymes that phosphorylate things or dephosphorylate them, depending on which direction they're working in. In our case, cyclin and CDK is responsible for the phosphorylation of RB, retinoblastoma. RB is something we call a tumor suppressor gene, and we'll talk about why here in just a second. RB is turned off when it's phosphorylated, and it's on when it's not phosphorylated. So you can see that what cyclin and CDK do is they turn RB off. They kill it. This is important because RB is a co-repressor for another transcription factor called E2F. Now, E2F is the transcription factor for a whole bunch of DNA enzymes. This is like DNA polymerase, ligase, all these enzymes that you need to make in order to do the cell cycle, to do cell division, DNA replication. E2F is a transcription factor that helps those move along. So we've been told that cyclins and CDKs help advance the cell cycle. Yes, they do. RB is a co-repressor for the thing that promotes DNA enzymes. If you say that this is E2F, and you say that E2F is going to sit on the enhancer, and it's going to help produce more DNA enzymes, what RB does is it covers it. This is RB. It says, don't do that. RNA polymerase tries to come in, and RB basically goes, go away. Don't express this gene. RB is the brake pedal for DNA enzymes. RB is the brake pedal for DNA replication. That's why it's inhibiting this transcription factor that helps DNA replication. Cyclins cut the brakes, right? Cyclin and CDK here, they're responsible for turning off RB. They are cutting the brakes. They're destroying this co-repressor, and this can continue on its merry way. That's why cyclin and CDK advance the cell cycle. They destroy the brakes. But what if you want it to break? What if you wanted to stop replication? You don't want to propagate cellular stress. You don't want to propagate mutations. You want to contain the damage. You don't want more DNA. Well, you want to stop this process. And if cyclin and CDK cut the brakes, you want to cut cyclin and CDK. This is precisely what P21 does. P21 will inhibit cyclin and CDK, meaning that this reaction, where RB gets turned off, never happens. And if this reaction never happens, because cyclin and CDK are off, this is always on, and it's always suppressing E2F. If E2F is always suppressed, this reaction doesn't happen, and these never get made. And you've successfully stopped the cell cycle. You've successfully stopped DNA replication. So, cyclin and CDK cut the brakes to DNA replication. P21 fixes those brakes. It puts RB back in business. Now, this is from cellular stress. What if you just have damage? Like you get a cut or a bruise. You need something that is going to help you heal, right? You need something that's going to cause healing to happen. Let's say you get a cut and you need to make new cells to fill that cut. A different pathway is taken. So this is more corresponding to DNA damage, and this is more corresponding to physical damage. Mike, or Mick, whatever you like, is activated in this case, and it is just a transcription factor for E2F. Remember, we said transcription factors upregulate. So Mike is the transcription factor for the gene that makes E2F, which means that E2F is made whenever Mike's around. Makes more of this. And if more of this is made, let's say this is not here because the cyclins are working, more of this is made, more of this is made. So Mick actually speeds up cell division. P53 slows down cell division, responding to different kinds of damage. Now, why is RB called the tumor suppressor gene? Well, tumors are just unregulated cell growth, right? You have a cell that's multiplying and multiplying and multiplying out of control. That's a tumor. If E2F is always on, and this is always running, you are always doing cell replication. If RB is off, you have a tumor, which is why RB is the tumor suppressor gene. It keeps E2F in check. It's the brake pedal. Cyclin and CDK, well, they just cut the brakes. P21 uncuts the brakes. So this is the entire cascade.